This is a walkthrough of the model for the gene regulation BioGraph activity. This is a model of or a simplified model of the lac operon in a prokaryotic cell. So I'll click Set up Gene with Repressor, and you can see the basic setup of this model. First, here's the interface. There are different buttons to set up a gene with a repressor, without a repressor, to add RNA polymerase, add lactose, and also to run the simulation for 40 or 200 seconds. There are some data boxes to report information about how this simulation is working and what the outcomes are. You can also see a graph of the lactose and the enzymes. And then this little green square or box, rectangle, is a result of a current bug in the StarLogo renderer, so you can safely ignore that. What we have here is this strip in the middle represents a strand of DNA, and the different colors denote different parts of that strand. So this purple part is the promoter, where the RNA polymerase will attach to the DNA strand. This green part is the operator. This purple pyramid is the repressor, and then this red part is the actual gene that codes for the beta galactosidase and the other enzymes, which we're just referring to as lactase. So let me set up, I already did gene with repressor. Gene without repressor just does not have a repressor bound to the operator. I can add RNA polymerase, and that shows up as these light blue diamonds, and I can run for 40. Oh, this castle is supposed to be a ribosome, and so you'll see RNA polymerase has started walking down the DNA, and it is transcribing that DNA, creating the mRNA, which goes up to the ribosome, and then the ribosome translates that and creates these Pac-Man shaped enzymes or proteins, which are the enzymes. Now if I do have the repressor and I add my RNA polymerase and run for 40, you'll see that the RNA polymerase is detached from the DNA strand and transcription does not happen. But if I add lactose, the lactose is capable of interacting with this repressor and will eventually, if the lactose interacts with this repressor, the repressor will no longer bind to the operator and the transcription will be able to proceed. Notice that what we're modeling here is the repressor mechanism and how the lactose interacts with that repressor and then enables the transcription to take place. The actual process of transcription and translation at the ribosome is just a visualization of that process, but we're not actually modeling the way that mRNA finds its way to a ribosome, or the way that the ribosome reads the mRNA and assembles the protein out of the component amino acids. And that's it for the gene regulation model.